Okay, so we're going to continue talking about the process of DNA converting into proteins, which involves transcription and translation. And then we're also going to talk about some other genetics, modes of inheritance. And we're going to start with talking about a geneticist named Thomas Hunt Morgan, who lived born in 1910, and he did experiments on the fruit fly Drosophila melanogaster. And he discovered a mutant male fly with with white eyes. And most of these flies have red eyes, but he found some that were male that were white. And so, to determine whether it was recessive or dominant, he did a cross with the red-eyed females, and he found that all the F1 progeny had red eyes. So, red was the dominant trait. When he crossed F1 females and F1 males, so the progeny from this red and white-eyed individuals, he found that he did find some white-eyed flies, but all the white-eyed flies were male. When he did a test cross with the F1 females, he did find that some of them would produce white eyes, but he deduced then from these experiments that the gene for eye color must be on the X chromosome, because flies have a similar sex determination in their genes as humans, in that they have males have both an X and a Y chromosome, and females have two X chromosomes. So in humans, we have 46 total chromosomes, 22 pairs of which are autosomal, which are not sex chromosomes, and then one pair of sex chromosomes, which are Y and X. The Y is a lot smaller than the X, and basically just codes for maleness, but there are a lot of necessary genes found on the X. There are some other systems, or birds, the females actually have the equivalent of the Y, but they call it a ZW rather than an XY. Grasshoppers, if they only have one X, they are male. If they have two X, they're female. And honeybees, if they're diploid or haploid, determines whether they are male or female. So what we call disorders or inheritance which are linked to the X chromosome, they are called sex-linked. Okay, and there are a few of these which have already been discovered to be sex-linked. One of them is red-green color blindness, which is more prevalent in males because it's found on the X chromosome, and there are some types of hemophilia also found on the X chromosome. So in these pictures, if you cannot see this sailboat here, you are probably red-green colorblind. If you cannot see the numbers here, which is a 71 or 74, then you are also probably red-green colorblind, which is more prevalent in males than females. Hemophilia, you can look at pedigrees and see how this is inherited. In the royal family, it was brought into, it was very common in the royal family because of Queen Victoria, who was a carrier, didn't have hemophilia, but carried the gene on her X chromosome and passed it to many others in her progeny. So what's kind of interesting about the X chromosome is in females and males, they're only using one. Males only have one X, so they only have one to use, but females have two X's, and what they do is they have one of those X chromosomes basically turned off or inactivated, and it highly condenses and becomes what's called the bar body. Now this isn't the same X chromosome throughout the body, and so females are basically a mosaic between these different X's that are turned on and off, and that's what accounts for the different coloring patterns in cats. One of the X chromosomes has a black color, and one of them has a brown color. There are also chromosomes, though, that fall outside of the things we've talked about, and those are found in chloroplasts and mitochondria, which have their own DNA, and is usually passed from the mother to the offspring, which would be maternal inheritance. Some systems, a father passes it, but for the most part, it comes from the mother. So you get your DNA from mitochondria through your mother. So one thing people have done with chromosomes is they mapped how close the certain genes, or the locations of the genes, or loci, are on the chromosomes. And they've done this based on genetic recombination events. If two genes are on the same chromosome, then they will generally be inherited together. But if they are crossed over, then they won't be. So you can determine in the progeny of, say, fruit flies, how close two genes are by the frequency of recombination. Alfred Sturtevant was the person who discovered this, and he was an undergrad in T.H. Morgan's lab. And he basically made a standard by which you could determine the length between different traits on the same chromosome. And genes that are further apart are more likely to be crossed over or on separate chromosomes in a crossing over event than genes that are closer together. And the distance then is proportional to these recombination events. This doesn't make sense. We're going to do some problems where it will. And he came up with a unit called a map unit, or also called a centimorgan, so named after his advisor, Thomas Hunt Morgan. So there are a couple different tests you can do. You can do a 3-point test cross, which is basically looking how close 3 genes are on a chromosome. And this is done by, again, looking at the progeny of these traits, or looking at the progeny at the traits at the progeny, the traits within the progeny. Okay, and if they are closer together, if the frequency are closer together, then you can determine they are close together on the chromosome. And we'll again do some of these in class. So we've done this with humans to determine a lot of genetic diseases, but it's very difficult because there aren't very many markers, not very dense, up until the 1980s, and a lot of disease-causing alleles are rare. So what they did is they developed what's called anonymous markers, and they are able to more finely determine where genes are. With the advent of new technology, we were also able to determine the difference in nucleotides, or single nucleotide polymorphisms. Polymorphism means different versions of the same thing. So, for example, we have here a DNA molecule where that you have the two sequences are exactly the same, except for one different nucleotide, and that's called a SNP, or single nucleotide polymorphism. And if you look at a bunch of these over a gene, you can determine paternal inheritance, for example, based on the similarity of these single nucleotide polymorphisms. Another disorder that was very important in determining things was the sickle cell anemia. It's the first human disease to shown to be the result of mutation in a protein. In the protein it was mutated was hemoglobin. What happens when you have sickle cell anemia is not all of your red blood cells are round. Some of them are sickle-shaped or have kind of a flattened, longer shape. This makes it harder for the blood cells to deliver oxygen, but it is also helpful in reducing malaria, because the protozoan, or the pathogen that infects blood cells that causes malaria, cannot infect these sickle-shaped cells. So heterozygotes for the sickle cell anemia will actually have a resistance to malaria. If you're homozygous for sickle cell anemia, though, there will be lots of complications. You usually have a reduced lifespan. So you see areas where there is lots of malaria. These are usually tropical areas, so tropical Africa, tropical India, and some other areas. Where malaria has been very prevalent, you will also see a higher degree of sickle cell anemia because of its advantageous for combating that disease. Well, we talked about this before, Nondisjunction. I'm just going to briefly mention it again. We talked about, it's a failure of chromosomes to separate evenly during meiosis. Aneuploidy is a gain or loss. If you have monosomy, that means you only have one when you're supposed to have two. Trisomy means you have three when you're only supposed to have two. And in most cases, especially these larger chromosomes, if you have trisomy or monosomy, then it's fatal and the baby or the zygote doesn't, the fetus doesn't develop. But on the smallest one, like trisomy 21, you are able to survive, and that would be with Down syndrome. Nondisjunction is when, or nondisjunction of sex chromosomes is another example where many of the trisomy or individuals are able to survive, and some of them are even able to reproduce. Triple X, XXX, if you have 3X chromosomes, it's called triple X females. If you have XXY, that's a male, and that's called Klinefelter syndrome. If you have XO, so only one X monosomy, you would have Turner syndrome. And if you only have the Y, though, because the X is necessary for life, you would not survive. XYY is called Jacob syndrome, and there are some problems associated with that as well. Genomic imprinting is also a common genetic chromosomal disorder, and this is basically where it depends on if you got a your mutation from the mother or the father will change the result in what disorder you have. So, for example, if you have a partial deletion of chromosome 15, if it comes from the father, it is Prader-Willi syndrome, and that's this here on the left, and if it comes from the mother, it is Engelmann syndrome. And these have some similarities, but for the most part are very unique diseases. Alright, so all these things, when we start talking about proteins and things that surround genes, around the genes, this is called part of the epigenome and is in a field of its own called epigenetics. So this is changes to DNA or the proteins around DNA, such as histones, that can be inherited. There's really no change in the DNA, but the interaction of the DNA, such as methylation, which can turn genes on and off, can happen. And you can see this in identical twins. They have the exact same DNA. When they're 3 years old, all the DNA is basically being used the same, but by the time they're 50, you see there are stark differences, and you see that in the different colors in the chromosomes, stark differences in how the DNA is being used, and that's because of the epigenome. So another way to detect genes, especially for developing fetuses, especially for people that are are prone to certain genetic diseases because of their mothers, grandfathers, and so on and so forth. You can look at the pedigree and determine, hey, based on what genes we think you have, this is the likelihood that your children will have this genetic disease. Or there are a couple procedures. Amniocentesis is where they take out some of the fetal cells from the amniotic fluid and then do genetic analysis on those. Or there's also chorionic villi sampling, where they take out cells from the placenta. Both of those contain cells provided by the baby, so you can do genetic analysis on those cells. The last, and this is kind of cool, but also has some ethical questions to it. It's called PreImplantation genetic diagnosis. This involves some of the steps of in vitro fertilization, where you take eggs out of a female and you fertilize it with the male's sperm, and then you let them develop outside the womb. That's what in vitro fertilization is. And when they're at a certain stage, you can actually take out one of the cells, do genetic analysis on them, and then determine which of those zygotes to implant. So some of them may have disease and you may not want to implant those. Or you can also do this for any other trait, so whether they're male or female, or genes for tallness. So there is some ethical questions around that. Alright, so this is the first part. We're going to continue and talk about transcription and translation on the next part.